I'm going to talk about pharmacogenetics and how we're integrating that into the electronic health record, specifically decision support. I'm not going to go through this in much detail. You know, there's a lot of terminology out there. Basically, these days, pharmacogenetics relates more to single gene variants that are associated with individuals' drug response, whereas and the three fields that it's used in at this point in time most intensively is oncology, cardiovascular disease, and psychiatry. Pharmacogenomics is based on whole genome analysis. William Fitzhugh talked about this earlier. In our experience, we're getting whole genome sequencing and analysis in the range of $1,000 to $5,000. I'm going to talk about sort of the poster child for pharmacogenetics, and that's Coumadin. It's prescribed about 2 million times to patients in the US every year. It's one of the top 4 drugs that cause adverse drug events. And it's a blood thinner. It's actually rat poison. And you can have anything from hemorrhage to clots, depending on your clinical and especially your genotype. The differential response between individuals is based partly on genetic heterogeneity. If you use just clinical factors, you get about 12 to 17% accuracy in terms of determination of the initial dose. If you add in genotypic information from the VCO, RC1, and CYP2C9 genes, you get an accuracy closer to 60%. We have a website that we've developed. I'm not going to go through this in much detail. You can go to it. It's called warfarindosing.org. It's got 26 algorithms that underlie it. It's been funded by the NIH. It goes into clinical things like drug interactions. And we're also working with, and I'll go into this, just mention briefly, one of 2 big prospective clinical trials to see how, to really confirm that genotyping is of tremendous use in warfarin dosing. Here's an example of the genotypic pull-down menu from the website. As you can see, for VCO RC1 up there, this patient has a GG signal nucleotide variant. They're warfarin insensitive. If you have a GA in that same position, you're warfarin sensitive, very warfarin sensitive. And this is not a metabolizer. This is actually the promoter region, which is about 1,600 base pairs up from the gene itself. And so it's actually a site where a protein binds to turn on the gene. And then we have this, obviously, a lot of cytochrome, CYP2C9, and other genes that are metabolizers that play a role in this. So this is all crunched together and comes out with a recommended dose, initial starting dose, and a dose from day 1 through day 10. We're doing now this through web service. This is a kind of simplified diagram. So we have a warfarin web service. We're using SSL technology, but we also have other layers of data protection. This is for electronic health records and also for physician order entry systems and other platforms. And this is being run out of Isodynamic LLC. And I'd like to end, I hope I did quickly. My major collaborators are Brian Gage, who's at Washington University, who's really the person that's doing this big clinical trial and has been sort of the intellectual driver of this project. Craig Robeson, who's done a lot of the web services work and hosting. Ken Kawamoto at Duke University, who's also involved in this. And then also I'd like to point out the vendors that are participating in this project. They include Zixi, which is MedStar's homegrown electronic health record, which was then commercialized by Microsoft Health Solutions to Amalga. GE Centricity, a bunch of different EHRs used at Washington University, and McKesson, which is used in the Duke University Health System. Thank you.